Hey everyone, so in this bio video I'm going to be explaining how the cell cycle is controlled by internal and external mechanisms and how if our bodies loses this control it could lead to cancer. So to start off I'm going to talk about the process of mitosis. Now what exactly is mitosis? Well, mitosis is a process of nuclear division that takes place in eukaryotic cells, and more specifically somatic cells, which are body cells in humans. It is divided into 5 stages: interphase, prophase, metaphase, anaphase, and telophase/cytokinesis. Here you can see a broad overview of all the stages that I'm going to be going more in depth into. The orange arrow points to interphase, the red points to prophase, yellow to metaphase, blue to anaphase, and green to telophase. So to start off, I'm going to talk about interphase. Interphase is when the nucleolus and nuclear envelope are still distinct and chromosomes are in the form of chromatin. In prophase, the chromosomes are condensed and the nuclear envelope is no longer intact. The cell then goes into metaphase, where the coiled chromosomes, each with 2 sister chromatids, are lined up along the metaphase plate, which is the dotted line on the cell diagram. The chromosomes are now at opposite poles of the cell, and metaphase is the longest stage of mitosis, lasting about 20 minutes in the total cell division process. Next is anaphase, which in contrast to metaphase is the shortest stage of mitosis. In this phase, the sister chromatids that are fused together to make the chromosome split and move towards opposite poles in the cell. Telophase then occurs, in which 2 daughter nuclei form and the nuclear envelope reforms. The division of cytoplasm occurs and the chromosomes become less condensed. This is where the 2 daughter cells appear, and cytokinesis involves the formation of a cleavage furrow, which is right in the middle that separates the cells in 2, and it sort of pinches the cells into 2 identical daughter cells, each with 46 chromosomes, just like the parent cell. The stages in this cell cycle are highly regulated and could have damaging or fatal mistakes if one is made. The cell cycle is also directed by internal checkpoints that are an operating set of molecules in the cell that trigger and coordinate key events. These checkpoints are the control points, where the stop and go signals regulate the cycle. The diagram shows an overview of the checkpoints. The 3 major ones are G1, G2, and M. The checkpoints receive signals from cellular surveillance mechanisms that report on whether crucial cellular processes have occurred properly and whether or not the cell cycle for that particular cell should continue. The cell cycle clock is also controlled by cyclins and cyclin-dependent kinases. Rhythmic fluctuations control the molecules that pace the sequential events of the cell cycle. These regulatory molecules are proteins of 2 types: protein kinases and cyclins. Protein kinases are enzymes that activate or inactivate other proteins. They give the go-ahead signals at the G1 and G2 checkpoints, right there. Many of the kinases that drive the cell cycle are constantly present but are inactive most times. Here you can see how when the kinase activity is low, the number of dividing cells goes up. In order for the kinases to be activated, it must be attached to a cyclin, another protein. Because it must be attached to a cyclin to be activated, the protein kinases is known as the cyclin-dependent kinases. The activity of the cyclin-dependent kinases, or the CDK, on the changes in the concentration of its cyclin partner is dependent on the changes in the concentration of its cyclin partner. In relation to the cyclin, the MPF, or the maturation promoting factor, activity directly correlates with the cyclin concentration. Its job is to trigger the cell's passage past the G2 checkpoint into the M phase. The fluctuation of MPH activity and cyclin concentration correlation can be seen in the chart. When the cyclin concentration goes up, so does the MPF activity. MPF is what is known as an internal and external signal, along with PDGF, which I'm going to explain next. The PDGF acronym stands for platelet-derived growth factor, which is made by blood cell fragments called platelets. The diagram illustrates the necessity of PDGF in the division of fibroblasts. So in the diagram, the very first petri dish contains a sample of human tissue that has been cut into smaller pieces. The enzymes are then put into the bottle to digest the extracellular matrix in the pieces, leaving behind the fibroblasts, which is the second step in the diagram. Things to make the cells grow, such as amino acids and glucose, are added into each of the two petri dishes that can be seen with the cells, but the one on the right has a PDGF also added. As can be observed, the cells in the petri dish on the left fail to divide while those on the right do. The reason this happens is because the cells have PDGF receptors in their plasma membranes, and when PDGF molecules bind with these receptors, they trigger a signal that allows cells to pass the G1 checkpoint and proceed with division. Now, say there is a loss of control in the cell cycle. What happens then? Well, in most cases, cancer develops as the form of a tumor. Cancer cells are cells that do not obey the normal signals that regulate the cell cycle. They divide excessively and evade the other tissues, maybe even the entire organism if left unchecked. In the two pictures, you can see the normal cells. Up top, you can see the normal cells and how nutrients, growth factors, etc. limit the cell density to only one layer. However, in the cancer cells, they continue to divide beyond a single layer, overlapping and clumping together, thus forming the tumor. The cancer cells can go on dividing indefinitely if they are given nutrients. Likewise, if the cells decide to stop dividing, the cancer cells that is, they do so at random times and not at checkpoints like normal cells. So that was it, and I hope it was helpful.